بسم الله الرحمن الرحيم. هبدأ معاكم إن شاء الله. I will begin with you with the course under the title Medical Bacteriology. I first like to introduce myself. My name is Dr. Amal Khalil, Professor of Microbiology and Immunology, Faculty of Pharmacy, of course, and Alexandria University. This course, Medical Bacteriology, is a bit huge. So we usually divide it into two parts, and I'm going to deal with part one. And my colleague, Professor Dr. Hoda Omar, will give you the part two, inshallah. Medical bacteriology part one includes gram-positive bacteria, acid-fast bacteria, and other miscellaneous bacteria. And at the end of the semester, I'm going to give you one lecture under the title Environmental Health. So don't forget that we are going to take also Environmental Health. First, we're going to start with a general introduction to medical bacteriology. Of course, our Earth, الأرض بتاعتنا, is full of bacteria. But of course, not all of these bacteria are pathogenic. يعني ما يقدروش ان هم يسببوا الامراض. ايه اللي بيدي ايه الكاركتر او الفينومينا، مش الفينومينا، الكاركتر بتاعت البكتيريا اللي بتديله القدره على ان هو يبقى ممرض او to cause a disease to the human beings. فالمicroorganisms that cause disease are called pathogens. Pathogenic microorganisms have special properties, زي ما قلت, that allow them to invade and infect the human body. But they have certain characters over other microorganisms that they can invade and infect the human body. Then it starts to secrete toxins and enzymes, which give the pathogenicity of the microorganisms, and it is manifested by a disease of certain symptoms and signs. أي disease of course أنا بقدر أعمل له أكشف عليه بأن هو بيظهر symptoms معينة و signs معينة للمرض ده. إيه الفرق بين كلمة symptoms? What is the difference between symptoms and signs? Symptoms are changes in the body function which are not apparent to an observer. Example, البين. هل أنت تقدر تشوف البين بتاع المريض؟ Of course not. Headache, malaise, يبقى دي كلها حاجات المريض بيشعر بيها but you can't observe it by your eye. Signs are very characteristic by changes occurring to the patient. These changes the physician can observe or anybody can observe and measure. زي lesions, swellings, paralysis, fever, etc. يبقى دي كلها حاجات you can see it by your eye. زي برضو rashes for instance, دي you can see it by the eye and you can measure it by certain instruments like the temperature. A syndrome is a group of signs and symptoms which accompany a particular disease. So we can هيقابلنا مثلا في الكورس بتاعنا حاجة زي مثلا Staphylococcus skin syndrome, scalded skin syndrome, toxic shock syndrome. يبقى دي بتبقى مجموعة من symptoms with signs together, so they call it syndrome. إنها دايما is accompanied by a certain disease. طب برضو في definition نحب نعرفه كلمة virulence. What is meant by virulence? Virulence is the degree of pathogenicity. That is to say, highly virulent organism exhibit pathogenicity when introduced into the body in very small numbers. يبقى إذا كلمة virulence دي بتوصف بيها البكتيريا أو الأورجانيزم. نقول this organism is highly virulent أو is have low virulence. دي بمعناها إيه؟ معناها إن very little number of the cells of this bacteria can cause the disease. فكل ما كان this number is very few, زي for example tuberculosis, Mycobacterium tuberculosis, it can occur by very little number of tubercle bacilli. And here we call it as a highly virulent organism. If we want to describe the disease, we can describe the disease by its severity and duration, for instance. So in here we are going to call the disease an acute disease, and this develops rapidly but lasts shortly. Example الانفلونزا. طبعا المعلومات دي ممكن تكونوا اخدتوها قبل كده في الباثولوجي او في اي ماده عندكم فكره عنها، فانا بعمل عمليه ايه refreshment for your minds في البدايه بتاعه الكورس. كلمه chronic disease طبعا كلنا عارفين يعني ايه chronic disease. It means develop slowly. The body's reaction is less severe but is continual. يعني ما بيحصلش ايه curing, complete curing ما بيحصلش, but it's continual, and recurrency is a occurs most common for long periods of time. بيحصل recurrency, ودي برضو زي مثلا tuberculosis. Subacute disease is intermediate between acute and chronic. Such an example we're going to deal with إن شاء الله برضو اللي هي subacute bacterial endocarditis. هناخدها مع streptococci. Latent disease هنا causative organisms remain inactive for a time. يفضل ممكن في الانكيوبيشن period of a certain disease فبيفضل latent الأورجانيزم، بعد كده بيبتدي يظهر and becomes active. ايه هي الميكروبيال mechanism of pathogenicity، او ممكن اسميها برضو I can call it aggressive mechanism of bacteria. Number 1 البورتال of entry. البكتيريا علشان تسبب a disease it must have the ability to enter the host body. According to كل نوع من البكتيريا it has a certain portal of entry. بمعنى ايه؟ يعني if it enters by another portal of entry it will not cause the disease. فندي هنا examples ونقول تاني نشرح الكلام ده. Many pathogens have a preferred portal of entry. If they gain access to the body by another portal, disease might not occur or usually doesn't occur. Example is Salmonella typhi. This organism is known to cause typhoid. Must enter through the oral route. That means لو دخلت عن طريق other route it will not be pathogenic. حاجة زي Streptococcus pneumoniae. It must be inhaled air droplets. تخش الجسم عن طريق air droplets. Clostridium tetani. This must enter via skin or parenteral route. يبقى إذا الشخص لو ingested Clostridium tetani مش هيظهر عليه أعراض التيتانوس، أو لو حصل دي تبقى حاجة very very rare، حاجة تبقى نادرة جدا إن واحد يجيله Clostridium tetani نتيجة أكل مثلا. لا، هو دايما بتيجي نتيجة skin abrasion في السكين، فتحة في السكين opening in the skin or through parenteral route. Second step in the aggression mechanism of bacteria is adherence. Once the pathogen gain entry to a host, all of them have some means of attacking themselves to the host tissue so that they are not cleared out from the body. ما يحصلش clearance of the organism from the body. So they have to have a way to adhere themselves inside the host tissue. The some has surface molecules called adhesins that bind specifically to surface receptors on the cell of certain host tissues. For example, Escherichia coli. Escherichia coli, which is known to be a normal flora of the large intestine, adheres there by certain adhesins found on their fimbriae, so they can persist in the large intestine. Another example or another way here, like Streptococcus mutans. Streptococcus mutans produce a certain substance called plaque. This plaque covers the tooth and enables the this streptococci, to adhere on the tooth. And then, of course, it can cause dental caries because this organism is the causative agent of dental caries. Number 3, Treponema pallidum. It has a hook in its tapering end. This hook also helps this organism to attack the host cell. And finally, bacterial biofilm formation. This is an exopolysaccharide matrix produced by different types of organisms has this ability to produce this exopolysaccharide matrix. So it covers the organisms, and by this way it also prevent the attack of the immune system and even of the antibiotics, so they can adhere at the place without being cleared away from the certain site in the body. Afterwards, antiphagocytic properties. يبقى الorganism entered, then adhered to certain receptors or by any way, and then it begins to protect itself from the immune system. ونمبر 1 من الimmune system اللي هي non-specific immune system الفاجوسايتوزس. ففي بقى properties للبكتيريا انها تبقى عندها antiphagocytic properties. زي ايه؟ انها تكون a capsule around its cell wall. Some bacteria make a glycocalyx material that forms capsules around their cell wall. This property increases the virulence of some species by resisting phagocytosis. Of course أكيد أخدتوا ده in your immunology program في الترم الأول إن الكبسول أولا بيبقى very smooth السيرفيس بتاعه فبيخلي الفاجوسايتس unable to engulf the organism. لدرجة إن if this organism loses its ability to form the capsule, the organism will lose its virulence. وبنطلق عليه في الحالة دي virulent. بنضيف كلمة حرف A على كلمة virulent فبتصبح عكس المعنى فمعناها virulent يعني that it means not virulent. Example Streptococcus pneumoniae, الهيموفيلس influenza والباسيلس anthracis. ودي طبعا كلها organisms we're going to deal with it إن شاء الله in our incoming lectures. برضو على حاجات بتساعد الأورجانيزم to protect itself from immune system في بيبقى certain components of the cell wall. زي إيه؟ Cell wall of certain bacteria contain chemical substance that contribute to virulence. Example عندنا Streptococcus pyogenes. على السيرفيس بتاعها بيبقى في حاجة اسمها M protein. دي بتبقى heat و acid resistant protein. فhelps the organism to adhere and also to resist phagocytosis. Number 2, the second example, Mycobacterium tuberculosis. Its cell wall contains waxes. These waxes اللي هي mycolic acid also resist phagocytosis. ودي برضو هندرسها بالتفصيل لما نيجي للمايكوبكتيريوم tuberculosis. Something very interesting, number 3, hiding the antigenic surface. This is something very interesting and very intelligent of the bacteria to do. بيعمل هنا البكتيريا بتعمل ايه؟ البكتيريا cell cover the surface of the bacterial cell with a component which is seen as self by the host phagocyte and the immune system. It covers itself with something similar to the same substance in our body, in the host body. فبالتالي الimmune system will not recognize it as a non-self فمش هيعمل لها it will not attack it and it will not cause phagocytosis. فهنا phagocytes cannot recognize the bacteria upon contact and even the possibility of opsonization by the antibodies to enhance this phagocytosis is minimized. طب ايه الexample للكلام ده؟ عندنا 3 examples. Pathogenic Staphylococcus aureus produce an enzyme called coagulase enzyme. This enzyme clots الفibrin on the bacterial surface. We all know fibrin ده هو اللي بيعمل التجلط اللي في جسمنا البلاد clot. فإذا لما يتغطى البكتيريا covers itself with this clot, the body will not recognize it as a foreign substance. Treponema pallidum binds a fibronectin to its surface. والفibronectin ده عبارة عن serum protein. فطبعا السيروم بروتين ده موجود في جسمنا فالجسم مش هيعتبره foreign substance. And finally group A streptococci برضو is able to synthesize a capsule. بدل ما يبقى معمول من الجلايوكاليكس it's made of hyaluronic acid. Hyaluronic acid ده موجود normally in our body as in the connective tissue. فبرضو when it covers itself with hyaluronic acid هتفقد الانتيجينيسيتي بتاعتها. يعني بمعنى آخر the body will not recognize it as an antigen. Next. Production of extracellular enzymes اللي احنا بنسميهم which are called exotoxins exoenzymes. في examples كتيرة قوي، بس في حاجة ممكن نلاحظها. We must recognize something that all the examples written here are gram-positive bacteria، وهتلاقوها في الكورس في الجزء الgram-positive bacteria أكتر. فبنلاقي إن الgram-positive bacteria generally can produce extracellular substances عن الgram-negative bacteria. هنلاقي معظم الgram-positive عندها قدرة أكتر to produce substances, enzymes or toxins outside the cell while it is still living. فمن ضمن الexamples الhyaluronidase and they also call it spreading factor. هنشرحها أكتر بالتفصيل in the later in our lectures. فدي بتبقى produced by Staphylococcus, Streptococcus and Clostridia species. الcoagulase احنا لسه we mentioned it لسه في الفقرة اللي فاتت، اللي هو بيحول الفايبرينوجين to fibrin فبيغطي البكتيريا with this fibrin. فالcoagulase ده برضه produced by Staph aureus، ده specifically Staph aureus. Bacterial kinases or fibrinolysins، يعني ساعات بنسميها kinases وساعات بنسميها fibrinolysins، دول dissolve البلاد clots. فبرضه ده produced by Streptococcus و Staphylococcus species. يعني the mechanism هنا is reversing the action of the coagulase. الcoagulase بتكون الفايبرين ولكن الفibrinolysins dissolves this fibrin. الcollagenase and lecithinases دي produced by the Clostridia species. Leucocidins دي destroys the white blood corpuscles produced by both Staph and Streptococcus species. And finally hemolysins which lyse RBCs ودي برضه also produced by Staphylococcus, Streptococcus and Clostridia species. Of course these are some examples. We're going يعني ممكن يبقى فيه أكتر من examples دي of course. Production of toxin. طبعا التوكسين عبارة عن إيه؟ These are poisonous substances that are produced by certain microorganisms. They are often the primary factor contributing to the pathogenicity. The term toxemia refers to the presence of the toxins in the blood. يبقى وجود التوكسين في البلد circulating in the blood دي بنسميها الحالة دي is toxemia. لو الأورجانيزم and the toxin are circulating in the blood we call it septicemia. البacterial toxin may be either endotoxin or exotoxin. This is a very important part. We have to differentiate between the endotoxins and the exotoxins. إيه الفرق بينهم؟ فهنا this table will illustrate this difference. أهم حاجة بتفرق بين الendotoxins والexotoxins. فأول حاجة نشوف properties chemical nature. Chemical nature of endotoxins, this is an integral part of the outer membrane of the cell wall which is called a lipopolysaccharide أو made of lipopolysaccharide. دي طبعا من دراستكم from your knowledge دي بتبقى موجودة فين في السيل wall of gram-negative bacteria. ولكن exotoxins these are protein which have enzymatic activity. These proteins are produced outside the cell while the cell is integral and living. So that's why we call it exotoxin. The protein is produced extracellularly. If we look to the size, lipopolysaccharide is much smaller in size, about molecular weight 10 kilodalton, while proteins are much bigger in size from 50 molecular weight from 50 to 1000 kilodaltons. Here this is mark هنا. في the next slide هنشرح how this protein is produced in the cell. Number 3, a relationship to the cell. احنا قلنا قبل كده ان اللايبوبوليساكرايد ده عبارة عن ايه؟ It's the outer membrane of the gram-negative bacteria. So it's an integral part of the cell of the gram-negative bacteria. But the exotoxin is an extracellular substance produced outside and it is diffusible outside the cell. Denatured by boiling. here this is a lipopolysaccharide, so it can withstand 100 degrees centigrade, so it is not affected by boiling. But the exotoxin, which is protein in nature, فطبعا يحصل لها denaturation by boiling. But here we say it's usually, because there are certain exceptions. We'll see في بعض الأحيان there is exceptions in ال exotoxin can withstand boiling. زي ما هنشوف في Staphylococcus enterotoxin for instance. Antigenicity, both are antigenic. But from your knowledge, of course, the protein is more antigenic than the polysaccharide. Formation of toxoid, and this is done by formalin. I can convert exotoxins to toxoid, which is having the same antigenicity of the toxin, but it is inactive. ولكن this process cannot be done with the endotoxin. وده لما بيتحول, when we can convert the toxin to toxoid, we can use it as a very successful vaccine. زي ما هنشوف في الدفتيريا وفي التيتانوس etc. الpotency. If we compare both potencies, we can see that here the exotoxins is highly potent or more relatively higher potency. هنا بنقول 1 microgram can cause the disease. And this shows ازاي ان هو highly potent. هنا we need more than 100 microgram so that it can cause the symptoms and the disease. Specificity, low degree, and here is a high degree of specificity, and here there is a mark برضو, and I'm going to نشرح specificity of these exotoxins في the next slide إن شاء الله على طول دلوقتي. فهنتكلم عن النقطة دي والنقطة دي في ال next slide. Enzymatic activity, هنا there is no enzymatic activity here, there is usually enzymatic activity. Pyrogenicity. Pyrogenicity, of course, دي خاصية is a character of the endotoxins. So of course you're going to say yes, it is pyrogenic. ولكن هنا ما قلناش no، لا قلنا occasionally، لأن هنلاقي فيه حالة very specific حاجة اسمها super antigens in gram positive exotoxins ممكن تكون also super antigens، ودي هنشرحها برضو in our later in our lectures. Here we said the exotoxins, as we said, they are protein in nature. So how can we produce the protein in any cell? It of course needs a gene. الجين هو اللي هيؤدي إلى protein synthesis. Of course من معلوماتكم اللي انتوا عارفينها. طب this gene هيكون موجود فين؟ يا إما هيكون موجود في الكروموسوم of the bacteria present، فبنقول إنها production may be by chromosomal mediated، أو this gene ممكن يكون في البلازميد فبيبقى plasmid mediated production، or is due to incorporation of prophage in bacterial chromosome, and this is called lysogeny. بفكركم لورا كده فترة درستوا انتوا في الجنرال مايكروبيولوجي اللايف سايكل اوف ذا فيروس اللي هو البكتيريوفاج، وكان ليه تو لايف سايكلز، لايتيك لايف سايكل ولايزوجينيك لايف سايكل. هنا بنتكلم عن اللايزوجينيك لايف سايكل، بيحصل ايه فيه؟ البروفاج بيبقى incorporated في البكتيريال كروموسوم and stays living، يعني يفضل البكتيريال سيل intact ما يحصلهاش lysis، بس بتفضل حاملة carrying the gene of the phage. This gene بقى ممكن يكون tox gene، هو اللي بيخلي البكتيريا تبقى قادرة enables the bacteria to produce the toxin, the exotoxin. If this prophage is not present in the bacterial cell will not be able to produce the toxin. ففي الحالة دي we call this bacteria is lysogenic in nature، يعني معناها it contains a prophage inside its chromosome. And this is very important لأنها هتمر علينا كتير في المحاضرات ونقول مثلا this bacteria is lysogenic. You have to understand what is meant by lysogenic, that it produces the toxin by the presence of prophage. طب قلنا برضو في التيبل اللي فات that the exotoxins is highly specific in its action. وفعلا بنلاقي ان كل exotoxin بيبقى ليها target معين in the host cell which it acts on it. And accordingly we divide them to 3 types. We can call the exotoxin is a cytotoxin, and in this case it kills the host cells or it affects their function, destroys their function. Or it can be a neurotoxin. We call it a neurotoxin. This will interfere with the normal impulse transmission. Or finally, it is an enterotoxin, and this affects cells lining the GIT. And usually it is الانتيروتوكسينز بتسبب مثلا الفود poisoning. These enterotoxins. طب عايزين ناخد بقى فكرة عن الميكانيزم of the endotoxins، أو اللي هي بنسميها we call it sepsis، أو بنسميها endotoxic shock. يبقى ليها كذا تسمية، كان call it endotoxin exotoxic shock أو sepsis. What happens in this case؟ وزي ما قلنا it is mainly in gram-negative bacteria أو it is in gram-negative bacteria. When bacteria is degraded by the phagocyte هيحصل destruction للبكتيريا، هيحصل لها lysis، فأجزاء منها parts of the membrane اللي هو السيل وول بتاعها هيتحول إلى blebs. membrane blebs يعني زي الفقاعات أو البابلز. very tiny particles. These particles will be filled with the endotoxin لأنها parts of the cell wall. So they are released in the surroundings. These blebs migrate to another macrophage or to endothelial cells, etc. in the body. This will elicit the release of mediators أو effectors اللي احنا عارفينها and you took it أكيد in immunology course. اللي هم ايه بقى these effectors؟ زي الانترلوكين 1، زي التيشو نيكروزيس فاكتور، زي البليتلت activating factor، زي النيتريك اوكسايد and others. دول are responsible بقى for the symptoms of the endotoxin like the fever, thrombosis and hypotension. And this زي ما قلت they characterize the sepsis. So we'll see now that these symptoms will be similar in all gram-negative bacteria. لأنهم due to what? Due to these effectors, not due actually to the bacteria itself. It's due to these effectors. فهنلاقي symptoms mostly for gram-negative bacteria are similar. مش هيبقى فيهم very characteristic signs for most gram-negative bacteria إلا بقى لو gram-negative bacteria برضو can produce an exotoxin. لأن ده وارد إن gram-negative bacteria produces exotoxins. بس generally الأفكت بتاع الاندوتوكسين بيبقى واحد similar to all gram-negative bacteria. طيب let's see the mechanism of each of these just to refresh your mind. يعني الانترلوكين 1 بيعمل ايه؟ ده هو اللي بيسبب الفيفر. فالفيفر interleukin 1 we call it endogenous pyrogen goes through the blood to the hypothalamus and induces release of the prostaglandins which recess the body thermostat at a higher temperature. وبالتالي usually في gram-negative infections بنلاقي فيه لازم fever. الميكانيزم of hypotension بيتم نتيجة ايه؟ نتيجة التيشو نيكروزيس فاكتور which damage البلاد capillaries. فبتخلي البلاد capillaries يبتدي يحصل لها permeability، بتزود permeability بتاعتها، increases its permeability leading to loss of large amount of fluid. فده طبعا بيؤدي إلى إيه؟ الدروب في البلاد بريشر. النيتريك أوكسايد الماكروفاجز بتفرزه to kill the microorganisms. بس لو حصل excess of the production of this nitric oxide, its effect as a vasodilator will be very strong and it would also will cause the drop in blood pressure. فfinally هيحصل نتيجة الهايبوتنشن هيحصل drop في blood pressure and shock. Examples of microorganisms causing this sepsis زي ما هتاخدوا التايفويد، المننجيكوكال meningitis and urinary tract infections. طب what about the mechanism of the exotoxins? We call them a classical AB subunit أو type AB bacterial exotoxins. هنا احنا قلنا إن these exotoxins are produced from the bacteria while it is still living. فالبكتيريوم adheres to any mucosal surface in the body. فالبكتيريا هنا مش هي اللي هيحصل لها diffusion. البكتيريا ثابتة stable in certain place in the body, in the throat, in the stomach, wherever it stays, and هيحصل لها adhesion اللي اتكلمنا عنها in a certain place. And then it begins to release its synthesized diffusible exotoxin. يبقى الاكزوتوكسين هو اللي هيعمل ايه diffusion into the blood in the body. اول ما بيبتدي يحصل له ريليز بيبقى an inactive form وبيسموه توكسينوجين. وبيسموه التوكسينوجين. فبيبقى عبارة عن A, B and C. بعد كده بيحصل cleavage in the extracellular space of the C portion and it will be converted to the active toxin اللي هو ال AB toxin. Why do they call it AB toxin? A, it is the toxic subunit أو ال active subunit, okay? And B is the binding subunit. And the next slide. ايه بقى الميكانيزم of entry into the target cells؟ mechanism of entry of ايه؟ of the exotoxin. في كذا طريقة ممكن بتتم العملية دي، فمن ضمنهم هنشرح لكم Receptor Mediated Endocytosis اللي هي RME is Receptor Mediated Endocytosis. The native toxin binds through its binding B subunit to its target cell. زي ما قلنا إن الإكزوتوكسين have a certain target cell. قد يكون a nerve cell، قد يكون any cell في مكان معين، liver cells, kidney cells, whatsoever. فكل toxin ال B subunit بتاعه can bind to a certain target in the body. So it goes to this target cell. ال B subunit بيقوم بالرول بتاعه اللي هو binding to the receptor. The toxin is internalized in the cell in a membrane enclosed vesicle called an endosome. يبقى هتشوفوا دلوقتي بعد ده هيبقى فيه فيديو المفروض تتفرجوا عليه ان شاء الله بيوري لكم الميكانيزم ده. اوكي. فبيكون اندوسوم يعني عمليه اندوسايتوزس، فيخش التوكسين A and B inside the cell in the endosome محاط بالاندوسوم. بعد كده بيبقى فيه hydrogen ions entering the endosome. It lowers the pH. This causes the AB subunits to separate. هيحصل انفصال separation of the two units من بعضها. بطريقة ما مش مفهومة somehow the B subunit affects the release of the A subunit from the endosome اللي هو enclosed inside the endosome ده to be released فين؟ في cell cytoplasm to exert its toxic effect اللي هي ممكن تكون أي enzymatic changes ممكن تسببها this A subunit. طب ال B subunit بيعمل إيه؟ It remains in the endosome and then it is recycled to the cell surface. بيرجع تاني يخرج بره الخلية خالص من داخل الخلية ويطلع بره تاني outside the cell. Examples for أمراض بيبقى فيها this kind of exotoxins التيتانوس، البوتوليزم، الدفتيريا and scarlet fever. This is the video. I'd like you to see it. Video number 1, mechanism of exotoxin. Now we want to know the reservoir of infection, or we can say the source where we get the infection. In definition, it is the place where the infectious agent survives and maintains its species through growth and multiplication. The most frequent reservoir is the human sources, and the human can be a patient. And if you are speaking about a patient, بيبقى فيه 3 stages for any patient. He may be in the period of incubation of the disease, during the incubation period of a disease, that he is, of course, the symptoms is not apparent yet, but he is infected and he can infect another person. The overhead disease اللي هي in the case when the patient is with all the symptoms appearing on him and he is known to have the disease. The convalescent period is after being cured. After curing, the organism may still be in his body and he may still be a source or reservoir of infection. B, contact carriers. Like for instance the medical staff, because they are in contact with patients, so they can easily be infected. Their immune system may be good, so they will not be a patient, but they have subclinical infection. So this can be only detectable by the production of specific antibodies. If we take a sample from their serum, we can detect the specific antibodies of this organism. The third source of human as a source of infection is his individual organism, his individual own organisms, or which is well known as normal flora or normal microbiota. This is a relationship between two living organisms, our body and the microorganisms. This relationship is known as symbiosis. This symbiosis may be in three forms. It may be commensalism, that is to say, one is benefited and the other is unaffected. This occurs, example, في organism اسمه الكوراين bacterium. It can live on our eye, on the surface of our eye, and it doesn't cause any harm to us. Mutualism, it benefits both sides. Example, Escherichia coli in the large intestine synthesize vitamins like vitamin K and some vitamin B. And of course, the intestine provides the bacteria with nutrients. The organisms may provide also the body with digestive enzymes, and this is very important and useful. Also, the normal flora can protect our body from other harmful microorganisms by the competition. يحصل competition between the normal flora and any outsider. When it enters any other microorganism which may be more harmful to our body, there will be competition for the receptors, for the binding sites, for the nutrients, and this will let the harmful microorganism to vanish out. We can get rid of it very easily. So this is a very important point, of course. Parasitism, only one is benefited at the expense of the other. طب احنا عايزين نعرف the distribution of the normal flora in our body. The normal flora are present in several parts of our body. بس مش كل حتة في our body فيها normal flora of course. يعني في مثلا blood مفروض يبقى sterile. البرين مفروض منطقة البرين ما يبقاش فيها مايكرو اورجانيزم. طب ايه الأماكن اللي بتبقى مركز فيها المايكرو اورجانيزمز أو النورمال فلورا؟ فنمبر 1 in the eye and the skin and the upper respiratory tract طبعا بيبقى فيها مايكرو اورجانيزم. example مثلا الستافيلوكوكال species. الجي اي تي هي اكتر مكان بيبقى فيه normal flora. فعندنا في الاورال cavity for instance السلايفا contains millions of cells. الstomach it is nearly sterile due to its high acidity except الacidophilic bacteria or that which secrete urease enzyme. لأن اليوريز enzyme بيكسر اليوريا بيديني carbon dioxide and ammonia. This is increases the pH فبيساعد it helps the bacteria to live in this media. About the small intestine بقى, small intestine is برضو nearly sterile due to its peristaltic movement. الموفمنت بتاعت small intestine is not suitable for microorganisms to adhere and to stay in this place. Large intestine بقى, it is the most rich part with microorganisms of normal flora. We can say that 40 to 50% of stool is bacterial cells. Mostly facultative or anaerobic enterobacters زي Escherichia coli, Clostridium perfringens, Candida albicans, Enterococcus faecalis, etc. يبقى في a big number of microorganisms can live, are normal habitant of our large intestine. Opportunistic microorganisms. These normal flora اللي احنا اتكلمنا عنها في بعض الأحيان بيتحولوا من الميوتياليزم to parasitism. امتى ده بيحصل؟ Under certain conditions, the relationship can be altered. For example, الإشيشيا coli اللي هي موجودة في زي ما اتفقنا في ال large intestine. لو حصل انها change the place اللي هي عايشة فيه and gain access to other body sites such as urinary bladder، فتؤدي في الحالة دي إلى إيه؟ urinary tract infections، وتعتبر من أكتر المايكرو اورجانيزمز اللي بتسبب urinary tract infections especially nosocomially في المستشفيات. طيب حاجة تانية، if the host is weakened or compromised by infection example الإيدز. AIDS احنا عارفين this virus اللي بيسبب الإيدز ال HIV virus بيقلل خالص المناعة بتاعت جسم الإنسان. So of course this weakened immunity بيخلي ال opportunistic organisms أو these organisms يتحولوا from the mutualism to parasitism. Another example, by the administration of certain drugs such as steroidal anti-inflammatory agents لأن دي برضو impair the host immunity, or antibiotics لأن antibiotics ممكن interfere with the balance, the normal balance of the normal flora. For example الانتيبيوتيك لو برو spectrum antibiotic ممكن kills كل البكتيريا جرام بوزيتيف جرام نيجاتيف الموجودة في الجي اي تي، ويفضل for instance الكانديدا، فيحصل كانديديازيس، بيحصل overgrowth of the candida. يبقى كل ده كنا we were speaking about the human source as reservoir of infection. نيجي بقى number 2 animal sources اللي احنا بنسميهم في الحالة دي zoonotic infections. طبعا في animals كتير قوي ممكن تنقل لنا الأمراض زي الكاتل، الدوجز، المونكيز، الراتس، البارتس etc. Inanimate sources اللي هي free living bacteria for instance زي السودوموناس which can live even in the water, can live in cetrimide solution. فالسودوموناس can live freely and can be a source of infection يجي من حاجة inanimate، حاجة مش حية. الanaerobic spores of course دول can survive in the soil for decades، ممكن يعيشوا عشرات السنين as a form of a spore, and they can infect human beings such as Clostridium tetani or Clostridium perfringens. وبكده we finished our first lecture. See you next time إن شاء الله.